The specific onset of multiple sclerosis is unknown, but there are multiple ways which the disease can progress. The complement cascade can play a role in the progression of MS. The central nervous system is typically considered an immune-privileged site, meaning immune cells do not regularly circulate through the central nervous system due to the presence of the blood-brain barrier. This means the central nervous system typically relies on the innate immune system, such as complement, to manage infection and maintain a normal environment. While complement proteins are normally produced by the liver, due to the restrictive nature of the blood-brain barrier, complement proteins are also produced by astrocytes, cells that contribute to the structure and environment of the brain, and microglia, the macrophages of the brain. Oligodendrocytes are susceptible to binding of complement proteins due to a deficiency in complement inhibitor expression. C1q can bind to oligodendrocytes, which can trigger the classical complement pathway. In situations involving brain injury, mannose-binding lectin and phycolins can bind to DAMPs from dying cells to initiate the lectin complement pathway. Both complement pathways converge at the formation of C3 convertase. C3 convertase results in C3b present on cell surfaces. Phagocytic cells then recognize C3b and phagocytose them. C3b can be further cleaved to produce C3d. C3d can then be recognized by B cells. C3 convertase also leads to formation of the MAC, as well as formation of C3a and C5a. C3a and C5a are chemoattractants that recruit phagocytic cells such as microglia to the site of damage. C3a and C5a also promote inflammation and cause macrophages to produce reactive oxygen species. The inflammatory environment causes damage to other central nervous system cells, causing the production of more DAMPs and continuing the cycle of inflammation. The inflammatory environment causes damage to the blood-brain barrier, and it can become leaky. This allows for more complement proteins, autoreactive T cells, and autoreactive B cells to enter the central nervous system. It has been shown that CD8-positive T cells are the predominant T cell type in MS lesions. MHC1 is highly expressed on inflammatory cells, neurons, and glia in MS lesions. The autoreactive T cells attack the myelin sheaths and continue promoting the inflammatory environment present in the brain. Autoreactive B cells can produce antibodies against myelin sheaths, which allow for recognition by phagocytic cells or can initiate the classical complement pathway. There has been some evidence suggesting that antibodies against Epstein-Barr virus, which causes infectious mononucleosis, display a high antigenetic similarity to myelin proteins and can contribute to the progression of the disease. It's also been found that inflammation persists in the central nervous system despite not having a leaky blood-brain barrier in progressive stages of the disease, leading to the belief that the blood-brain barrier can be repaired and inflammation can continue behind a closed-off central nervous system. This can occur due to the formation of lymphatic-like tissue compartments in the brain that resemble B-cell follicles. B cells, plasma cells, and dendritic cells have been found in these dense B-cell follicle clusters and are widely present in patients with severe MS. Overall, the damage to myelin sheaths is what causes progression of the disease.